My name is Arnold MUNNICH. I am a pediatric geneticist, and I am the current president of the Imagine Institute for Genetic Disease at Necker Hospital in Paris. I am unable to attend in person as I am presently at the European Parliament in Strasbourg in an extremely stressful and frightening context for the future of Europe. And last year, I remember that we were in Jerusalem, where also the country is going through extremely concerning days and months. And in a sense, it is an appropriate time to exchange on where we doctors and scientists are embarking our humanity. And today we have an extremely interesting and relevant panel of speakers. The first speaker will be William MEZZANOTTE, who is the head of a very well-known company, big pharma company, CSL. Then the second one would be Giuseppe Novelli, my quasi-genetic twin, director of the medical genetic lab in Roma Catholic University. And eventually, Michaela SEMERARO, who is the head of the Clinical Investigation Center at Imagine Institute at Necker. And Michaela is a pioneer in the successful gene therapy trials in humans. And I'm very happy to have been able to bring together these three speakers. And I suggest that we discuss the presentation after each talk and to conclude in 1 hour from now. So the first speaker is William. Thank you, William, for being with us. And the floor is yours.
Thank you, Arnold. Can you hear me okay?
Yes, perfect.
All right, thank you. So I'm William MEZZANOTTE, and as Giuseppe told me 2 minutes before I started, I had the wrong name badge on. I had the name of some other Italian man with an M. So I am actually MEZZANOTTE. I'm from CSL. We are one of the largest companies that most people have never heard of, but 2 years ago, started to hear from us when we approved and delivered to the market the first gene therapy for hemophilia B, which is up for the Prix Galien International Award tonight for Best Rare Disease. So if any of you still have a vote, I'd appreciate your vote. Anyway, I was asked by Arnold to talk about democratizing gene therapy as a broad topic rather than the development process alone. So gene therapy research, of course, has been going on for 50 years. And since I started in medicine almost 40 years ago, there's been this promise of a new dawn of treatment for people with rare and serious diseases through gene therapy. And I was promised that it was right around the corner. And of course, that corner has continued to move, but it looks like we're finally almost there. And there's over 2,500 active clinical trials right now utilizing some form of cell and gene therapy. And over the next 5 years, the market in cell and gene therapy is estimated to quadruple in size. So that's the kind of expansion we're seeing rapidly. And this emerging wave of single treatment therapies is poised to make a significant clinical impact in the near future. And I am often quoted as saying that R&D in organizations like myself is in the business of hope. And really, nothing speaks of hope more than gene therapy for some of these incurable diseases. And I'll use, through this talk, our own example of hemophilia B, but I want to expand that much larger because the issue is the same across the spectrum of diseases and therapies that are out there. But people with hemophilia B live with the knowledge that they're vulnerable at any time to spontaneous bleeding or bleeding associated with trauma, leading to serious joint and tissue bleeding, injury, and damage, or even worse, life-threatening intracranial bleeds. And even when they're stable, constant, regular intravenous infusions reminds them really that of this vulnerability and the fact that they have a disease. Imagine what that would be like if you were free of that because you're making your own factor IX and that you no longer had to take treatment. And in fact, the Institute for Clinical and Economic Review, a U.S. body that looks at health effectiveness, looked at our data and predicted that our hemogenics molecule would remain effective for almost 23 years. So remember, that is the principle of science delivering great innovation for patients, really life-changing outcomes. I met a patient with hemophilia B that was in our phase 3 clinical trial just a few months ago. He got treated almost 4 years ago and has been followed up since then. And he has yet to have to take any prophylactic therapy since then, 4 years. And he asked me a really interesting question that I'll ask you. Am I really still a patient? It's a really interesting question. And it's apparently lots of psychosocial issues associated with that we should not take for granted. So with such ability, why aren't we making better and quicker progress in this area? Well, there's a lot of barriers. The complexity of medicine development in general, patents, regulatory frameworks, manufacturing efficiency, health system acceptance, that's a big one, including the payers, the health technology assessments, and the practitioners themselves. Of course, everyone wants to focus on cost, and cost is one, and we'll touch on that, and then finding cost-effective ways for the truly ultra rare gene therapies, which are really a whole conundrum themselves. And these barriers at times seem insurmountable, and some days I just wonder, but I think we're actually making progress on a lot of them, and I'd like to enumerate some of them in the next few minutes. So how about medicine development? Well, we're at a pivotal moment in healthcare with these new technologies, with these single-use therapies, but developing these therapies remains complex and challenging. For instance, the initial laboratory work for hemogenics started right around 2000, but the academic research started in the early 1980s. So that's almost 40 years of research to get to one therapy. Imagine if we have to do that for every gene therapy that's coming. We'll be waiting a long time for treatment for some of these diseases. And a successful new medicine is rare in itself. And it takes almost 15, 20 years from the start to the finish, as I just said, to move from discovery to market. And cost, if you put all in costs for other failures, for the infrastructure costs in R&D to support it, to the other associated costs in an organization, the most recent estimate is almost $2 billion per success. So that's a lot of cost in the system as well. And so we'll be waiting a long time if all that remains the same. So the fact of the matter is that identifying the correct viral vector, finding the gene cargo correctly is really difficult. And it seems simple in the lab, but it's not simple at all. And of course, right now, most of the gene therapies target the liver, which is a fine organ for factor IX, but is not a perfect organ for some of the other gene therapies we're considering. And so getting to the true target organ of interest is probably the next frontier. And we're not there yet. But I think when we start to get there, we may see a faster success in some of these areas. In addition, the clinical manufacturing is enormously complex. And I have some of my colleagues who've been involved in the clinical manufacture here in the room today. And I will say that it's each and every time, it's an individual process. And so when you think about a new disease and a new gene therapy, you're starting with a new vector sometimes, a new cargo, and a whole new clinical manufacturing process. And so you're starting from square one each and every time, which makes the process and the delivery painstakingly slow. And the regulatory pathway to approval is still being paved right now as well. Requirements can be quite cumbersome, can be variable, so you don't learn very well from one program to the next, and can be sometimes difficult to understand. And that's painful for a company like CSL that's large and has a lot of experience. But if you think about small companies or universities trying to do this, it becomes really difficult to do. And so finding a better way and a better framework to doing so will really help companies. And therefore, we have to reduce this complexity, and we're really excited because the NIH has a program called Foundation for National Institute of Health Progress on Gene Therapy. And they're trying to accelerate rare diseases. And we're excited about this because they're put together a playbook for standardizing gene therapy submissions intended to treat rare diseases, which affect populations too small to really be done effectively by any one company. And the playbook has streamlined templates, master regulatory files, uniform manufacturing, which will help. And in addition, they're doing a second pilot on gene delivery systems and manufacturing methods, almost so that you can repeat these ad nauseum for each and every new gene therapy. For some of the very rare and for some small companies and universities, this would be an enormous improvement. And so we await the results of that pilot. They plan on publishing all the results. And so this will be accessible to the whole world when it comes. So we applaud that. Another interesting area is patents. That universities and companies are often patenting both aspects of the genome as well as very small process improvements. Things that on one hand you think they deserve to patent because they've discovered it, but on the other hand, it does make it hard to follow that on innovation and improve the innovation, or costly to bring that innovation to the market. It's true even in our hemophilia product. We're using a variant of factor IX gene called the Padua variant, and that has been patented, and we have rights to that patent. But it is, it's in itself a challenge for others. So I think we have to undertake a cautious and rational approach to patents. Obviously, as a part of a company that is in the innovation business, we have to be able to reward innovation with protection. But on the other hand, we have to create rational approaches to that protection so that follow-on innovation, which is so critical to the way medicine and science works, is allowed to progress. And I think we have to be somewhat more thoughtful in that area. How about manufacturing efficiency? Well, right now, I think there's a big opportunity to reduce costs. The supply chain for gene therapy is highly individualized, choppy, and prone to disruptions from loss of key personnel. You lose 2 people in an organization, you lose your whole understanding of how to do that process right now. And often subpar constituents or funding to do so. So I think getting this supply better for patients and more reliable will make the decision to choose gene therapy even easier and for health systems. And we're enhancing process improvements, looking at impeccable quality and yield at CSL to try to overcome this issue. And I know others are doing it as well. How about the health system? Greater assuredness there would really help. So innovations can only be successful if the system that surrounds them is ready to support their uptake. And this entails that healthcare systems are integrated and prepared to identify eligible patients, administer the therapy, and continue the long-term care and follow-up needed. So we need to invest in building up this expertise and infrastructure. It's actually not there in many places right now. Therapeutic inertia is actually a real issue, and I've seen it in live and in person. While most rare diseases, there's substantial unmet need, the practitioners still remain to be convinced often. So clear benefit risk advantages over standard of care currently, long-term safety assurance, durability of treatment, and confident identification of the right patient for your therapy is going to be really important. We've now generated and published very favorable safety and long-term data over 3 years to help reassure the practitioners of our therapy, but for some of them, I think they want even more data. And then access is an incredibly important thing to the proper payer health technology assessments. In the US, once a physician decides that gene therapy is the right for the patient, the challenge is just beginning. For CSL, the patient, and the physician, there's a lot more work to do to justify the cost, and it often takes another year to make that decision and get that therapy in. In Europe, I'm hopeful that once countries complete their health technology assessments, which are undergoing right now, the system may be a little more streamlined, but that remains to be seen. And we're running that experiment right now because still people want to understand that the therapy is worth it. The other essential component here, of course, is the ability to come up with innovative contracting models which meaningfully help payers and governments manage the budget impact of the therapies. It's not really all in the hands of the sponsor or the manufacturer, which people like to believe. We need active policy changes to enable solutions. We're concerned about restricted funding, legal and administrative hurdles to enable the kind of innovative contracting solutions that may be needed. But we're at the same time encouraged by some governments who are sending a good signal of how they may think innovatively and collaboratively about this. So of course everybody wants to talk about cost, so finally cost. Making cell and gene therapies affordable and easy to get, overcoming financial and regulatory barriers is still very complex. So a single dose of HEMGENIX can generate significant cost savings for the overall health care system, since in the United States, the lifetime cost of somebody with hemophilia B is almost $20 million. HEMGENIX does this by reducing annual bleed rates, eliminating prophylactic therapy like the person I mentioned, avoiding hospitalizations and surgeries. The cost of HEMGENIX reflects these healthcare savings, substantially lowering the overall burden. The same is true for other gene therapies, which substantially reduce those burdens and costs. However, the cost of the therapy is front-loaded. And so because it's front-loaded, the upfront cost will always seem a bit of a sticker shock. And so we'll see much higher than those therapies and the yearly cost of a standard of care, even if the overall burden is less. So despite the obvious positive 10-year economic equation, the near-term economic equation makes payers and governments nervous. We're trying to educate the system of the true overall savings, helping doctors and patients with benefit issues, and also offering creative value-based agreements for commercial and government payers who are interested in that model to help reduce the risk to payers. Other payment models may emerge. Through these agreements, CSL stands behind the promise of HEMGENIX and the durability of the product in a way that can make it easily managed by payers using claims data and pay-over-time models. But of course, improving all the complexity that I mentioned would also help the cost as well. Finally, we're looking at cost-effective ways into truly ultra-rare diseases where clinical trials just will never be possible in the standard evidence that you want. And the NIH has a second ultra-rare disease initiative going on to provide funding and resources to advance gene-based therapies for ultra-rare conditions. And we note right now that the definition of ultra-rare is a little bit loose, but this pilot will be enormously helpful, I think, in the future. So really, in conclusion, scientific breakthroughs only become transformational when they reach the end patient, and that's why it's paramount that we use a community of scientific and business leaders and continue to democratize gene therapy, working with a sense of urgency. So reducing complexity of developing, rationalizing patents, improving the manufacturing and regulatory landscaping, streamlining health system acceptance, creative pricing models, and further cost-effective measures will go a long way towards our goal. So together, I think we can get there as a community. Thank you.
Very much.
Arnold, did you want me to answer a question now or go on to the next speaker?
Yes, we have time for a couple of questions. So go ahead. But first of all, let me thank you for your thoughtful, cautious, and wise approach of this very sensitive problem. But we have time for one or two questions. So stay on the floor, please. One question, one or two questions.
No hands so far, Arnold.
Okay,
Perhaps we'll come back to the discussion.
Yeah, you come back for the general discussion.
Thank you.
Thank you. Thank you. Congratulations, William. So I'm happy to welcome my colleague and friend, Giuseppe Novelli, a very well-known and famous geneticist, director of the Medical Genetic Laboratory at the Catholic University of Roma. And it's a privilege to have you, Giuseppe. We have known each other for very long. I am really eager to hear your thoughts about this question in general and DNA editing in particular. Thank you, Giuseppe.
Thank you, Arnold. Thank you very much. Thank you for organizing, for inviting me to this important meeting. The progress in science depends, of course, by technique, knowledge, and money. Thank you, Williams, for the speech about the money in this field. Gene therapy, finally here we are. Why? Because we spent the last 30 years in order to understand what we have up and down in this field of science. Recently, in the last few years, we have very important success in terms of gene therapy. For examples, one is the ocular diseases, in which we have now the gene therapy is completely changed some rare disease for ocular system. Another aspect concern the deafness, the congenital deafness of humans, in which you have many experience now with the first patient treated for congenital deafness in US, in Europe, in Israel at the moment. And another aspect, for me, it's the most important success of this disorder is that we have now cure for sickle cell disease. This is the most important, the very old classical genetic disease, because it's been described in 1949 by Professor Pauling, in which you know that it's a single gene, single mutation, single amino acids changed. And now we have the first patients at home completely healthy and completely free from these very dangerous diseases. And the same approach has been tested in the last few months for thalassemia and in Europe, in Italy, and in other parts of the world in order to treat these very important blood disorders. The scheme is the same. What is interesting is that for the first time, we not correct the gene affected, but we modify another gene in order to balance the production of the fetal hemoglobin that will be shut off at the birth. But using this approach, now we can active the gene in order to produce fetal hemoglobin that can be useful for these patients. It is very, 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 very important for the patient. But please let me a personal consideration. In the 1990s, I'm involved with this Arnold now very well in the mapping the locus for spinal muscular atrophy on chromosome 5. The gene has been cloned by Arnold, MUNNICH, Judith Melki in France. But what is important, I never find that after 10 years, we have now a drug for these patients. It is very impressive to look at patients affected by the classical and severe form of this disorder stand up after sub-administration of this drug, an RNA drug. This is fantastic for me. After 10 years after the cloning of the gene. This is very important success. We have here just a series of rare disorders in which we have now a drug for gene therapy using different approach. Now where many others are in trials at the moment, but this is results of the technique. How many techniques we have at the moment for gene therapy? In the last 30 years, we have knockout, no gene, gene correction, RNA editing, single base editing, transcription regulators, and others. We have a large number of tools to correct gene in the genome. But as a disruptive technology, we have good news and bad news. What about good news? Promising advance during the last 3 decades. We have the recombinant DNA technology. Bad news is that we have sometimes difficulty in efficacy for specific disorders, and sometimes we have difficult to understand the biological process in some particular cells or particular tissues or others. And the last problem is the off-targets. Of course, the off-target, this is an important problem for the recent gene editing approach. But what is important in the last 10 years, with what has changed, is the precision in which we can perform a modification of a single base in the large genome, like a humans, for example. And this is important because we have in the last 10 years, the precision of genetic engineering increased of about 10 million-fold. Can be compared to precision is equivalent to changing a single character in 1,000 books. It's very, very, very impressive in terms of precision. but the most important technique we use at the moment for gene therapy is the CRISPR-Cas9 technology of gene editing. This is, you know, it's discovered about 5-10 years ago now. It's very, very important people. And because this technique is able to perform gene disruption, gene correction, gene insertion. And this was initially tested in food animals farm, because it's important, because in 2017 FDA approved the use of this technology for modify the genome of this particular in food animal farm in particular. And after this, many other animals has been treated using gene therapy. Now it's a very common process in order to understand or to increase the production of some important food, of important milk, for example, or muscle or others. But now it's very, very routine in this field. But what about the humans? The genome edited by CRISPR is very important, because we use some particular protein enzyme in order to cut and join some very small piece of DNA. It's what is important. The modification is permanent at DNA level. It can be performed deletion, replacement, insertion of single base, and the approach is very fast and low cost. How many about the limits? No limits. This technique has shown no limits at the moment. One of the problems, the most important problem at the present is this, is the off-target effect of CRISPR-Cas9 in genome editing. This is a consequence of sometimes, because we have some problems in order to identify the correct base pair to change or to modify, and the scissor is not exactly recognized using the guide of RNA in order to identify the locus to be modified. However, in the last 3-4 years, many people developed technical approach in order to increase the efficiency and reduce the off-target situation. How? Modifying the RNA guide, using a new enzyme different from a new type of Cas9, is the first isolated, but we have other proteins at the moment, and improved some other aspects in terms of kinetics and others. But we have another problem in the human genome when we use this technique. This concerns the fact that the coding variants are often have pleiotropic effects across multiple tissues. For example, we have a gene that has a specific expression in the brain, but difference in the liver, for example. And it's not dependent by the gene, by the promoter or modifier genes or other genes that interaction with this. And this can be a problem when we need to correct the expression of a gene in a specific tissue or in a specific cells. Another aspect is the genetic architecture of the chromatin, what the gene is located, in which chromosome, which is the chromatin and the epigenetics effect of this locus. That's been difficult to reach, to achieve the gene to be corrected. This is one of the problems that we have. Another aspect is concerned about the ethical aspects, in particular use the gene editing in the germinal genome. This will be discussed by Michaela, therefore I will skip immediately this, but I have just a slide about this history that Michaela will be describing in details. It concerns the technique used by Ho Hyang-Ki has been a success or not in terms of biological. Please look at this free statement. In one of the two twins he modified, CRISPR modified only one of the two chromosomes because you have an heterozygous. The idea of the scientist was to modify both alleles in the genome. Therefore, this is a problem. The second is that he is not all the cells of these twins have the same modification. In other words, we have a mosaic of the results. All the results have been obtained after birth. After birth, when we can have the possibility to look at the results of these two girls. And other aspects concern the possibility that this germline's modification is permanent in other generations, of course. But this will be. What is in generally by this editing? What we have, of course, we depend on the vectors. Lentiviruses at the moment are the most efficient vectors to transfer these tools inside the cells. We have also enzyme, and of course, for each one of this technique, we have, of course, good and bad news. It depends on the technique you will be used. Another aspect that is to be important to understanding in this aspect is the biology of the cell, because cells are different. You can perform gene editing in stem cells. You have a different results in somatic differentiation cells. It depends, of course, because the status of the activity of the cell is different, they change. But therefore, we need to perform many experiments in many types of cells in order to reach the final of the efficacy of this technique, the other aspect to it. But what about the last few slides? The RNA world, which is the situation at the moment, because we look at the gene as a DNA, but now we have the possibility to look at the product of the gene, the RNA, and therefore we have many types of RNA. RNA coding for protein, non-coding RNA. A non-coding RNA represents 98% of the genome, because the coding part of the genome is only 2%. Therefore, the dark genome, it's very large, and it's important. Now we have different species of RNA that can be used for gene therapy approach. And if you look at this market situation now, as a manufacturer of RNA in the last few years, we see an increase of the product of drugs using RNA as a drug. And what about RNA therapeutics in terms of innovation landscape? In the last many clinical trials are at the moment are training in U.S. and Europe in very large group of disorders, but not including for the vaccine RNA, because it's just a different. Just to look at diseases of genetics and rare diseases or chronic or complex disorders like atherosclerosis and others now are very active. And what is important in terms of money, in the last, it has been estimated that the therapeutics market is projected to grow from 14 billion in the last year, reach to 18 billion in 2028. And this is what is expected. Only RNA. RNA is very versatile. This is another approach that we can use in my lab in order to use microRNA, this mRNA, in order to differentiate cells infected by a virus and cell not. Because if you have, for example, SARS-CoV-2 infection for the COVID, of course you have cells positive and cells negative. But what is important is if you can generate a drug in order to kill the virus inside the cells. You need a system in order to differentiate cells positive, infected, from not positive. And what we found is if you look at the profile of a microRNA, you can select and kill selectively cells infected from the other cells not infected, using just an RNA. This is a project founded by Italian National Ministry of Research. What is the last slide is what we have in the next situation. What about the gene replacement that started in 1995, but the last we have the gene editing, and now what is very recent is the epigenetic editing. This is a very new field for gene therapy because we look not at the gene, not at RNA, but the modification, epigenetics modification of DNA, for example, to change the methylation status of DNA. This is a new approach for many important disorders like cancer, for example. I would like just to stop with this slide because it's very interesting. This was stated by Hollentrump, as the editor of Science in editorial last year, which we have two lives in two wars. One of discovery and order, and the other one. And the other word is like of division. And this is very dangerous for us because we have fantastic technique in order to save the life of millions of people at the moment. Thank you very much.
Thank you. Thank you, Giuseppe. Thank you very much, Giuseppe, for this very nice, elegant overview. I suggest that we can take one or two questions because we kept your talk in time, so it's fine. Who want to address the first two questions?
Thank you very much. So, Chris Henschel from the Cube Labs team. One simple question and one slightly tough question, which I hope you accept in the spirit that it's given, because it's an inquiring mind question, not a hostile question. So the simple question is CSL originally the Commonwealth Serum Labs in Australia? Because I visited it so long ago that even I was young then, and it was just doing flu vaccines and very conventional vaccine work. And how on earth did you move from doing that to gene therapy? It seems like such a huge leap that I can't understand how it happened. That's the gentle question. The slightly less gentle question is, I was working in Philadelphia when there was what was supposed to be the first gene therapy trial. And I remember it very, very well, because in phase 1, the patient who was first treated died. As I recall, I don't remember the whole story, but it was somebody who was immunocompromised, and whatever virus vector was used got out of control, and this student didn't survive. Now that must have been such a blow to this whole field that I wonder how many years you think that put the field back.
Thank you very much for this question, sir. I suggest that we keep this question for the general discussion after having heard Michaela SEMERARO. This is a very relevant question for the general discussion. We keep this question for the discussion. Okay. So Michaela, thank you for coming. Michaela is the head of the Clinical Investigation Center at Imaging Institute. She is in charge of recruitment and treatment of patients involved in gene therapy program. So she is particularly relevant and aware of the question that you have raised, sir, that is the adverse effect of gene therapy. So Michaela, thank you for coming, and thank you for enlightening our reflection and our discussion today.
Good afternoon. Thank you, Professor MUNNICH, and thank you to the Galien Foundation for this invitation. As Professor MUNNICH told, I will discuss with you some ethical consideration in gene therapy trials. Just to remind you, gene therapy is a medical procedure where genes represent the medical product. And with the growing number of gene treatments available on market, it's important to remind that this intervention can have a profound and long-lasting impact. In this way, we have not to forget our principle as a caregiver and asking always what is good and what is bad. As told before, sometimes science seems science fiction, and science became like a movie a few years ago, in 2018, when a scientist in China was arrested because on gene therapy manipulation on embryos in order to try to keep them immune to HIV. This scientist went to jail for 3 years, and at this moment, the scientific community, but not only in the scientific community, through in a dilemma, what is ethical and what is not. But let me remind you what is gene therapy and why this technique is so important. Gene therapy is a revolutionary approach in medicine which modifies a person's genes to treat or cure a disease. There are three major points to retain on gene therapy. Gene therapy is a precision medicine, is a tailored medicine because we modify the sick gene in the patients. There are potential benefits at the biological level because the gene is corrected, but also in the quality of life of the patients. And last but not least, the gene therapy offers hope for previously untreatable diseases. For the gene therapy techniques, like Professor Novelli explained before, we can use different tools. We have here there is an example for hematopoietic stem cell transplantation, gene therapy on HSC. But in this case, we can use ex vivo gene therapy. So we manipulate the stem cell, in this case, outside of the body and then inject them into the body. And or in vivo gene therapy when we deliver the gene treatment directly into the body of the patients. And these two techniques utilize the gene addition if we apport the gene, or gene editing if we modify the genome. In these fields, we have different tools like viral vectors which are mostly AAV, like in hemophilia, or lentivirus, to bring the gene, the therapeutic gene, into the cells, or genome editing strategies to modify the genome, or mRNA to increase the production of defective protein in the sick cells. But the discussion about ethical concerns in gene treatments started several years ago, as here demonstrated in the act of the conference of 1966 of the group of scientists discussing on recombinant DNA. At that time, the question was, should scientists limit their responsibility in this area to technical concern, and how much is okay? During my talk, I will not talk about germinal or embryonal gene therapy or gene therapy for enhancing human traits, because these are some specific topics deserve to be discussed in a deep discussion. But I would like to talk with you about some fundamental ethical principle and giving you some example of my practical clinics. And the first is the beneficence, which means acting in the best interest of the patient. For example, I'm talking again on spinal muscular atrophy, because we know that there is a terrible genetic disease which affects very young people. And since 2018, there is a new gene therapy which is Zolgensma, produced by Novartis, which modified completely the history of these patients, since many patients with a single infusion of the product experienced significant improvements in muscle strength and in motor function. But there are also some ethical concerns to be discussed with the use of Zolgensma, because we know that SMA is a disease with different expression across patients, because there are some patients with a severe disease, with a life-threatening severe disease. And treating these patients means that this patient will be with a very severe handicap of all life long. And so we have to ask which benefit, which kind of life for severe SMA presentation. This is a very ethical issue, and in this case we have to consider that the communication with the patients, in this case with parents, because the patients are newborn or very young babies, it's very important. Moreover, because this treatment is not free of severe adverse event, like severe liver and renal toxicity. So parents need to be informed about all these possible events. how we can overcome this problem? Probably targeting the right population. Even because during the SPRINT study, it has been demonstrated that the patients treated pre-symptomatically show a rapid improvement in motor function. So we have to target preferentially patients with SMA with non-clinical signs of disease, because this, the history after treatment of these patients is very encouraging. For this aim of target population, different countries in Europe, but not only in Europe, decided to perform screening programs in order to detect SMA patients without clinical symptoms and define the eligibility of these patients to gene therapy. For instance, in France, we have the DEPISMA clinical trial with the aim to identify this kind of patients and discuss at a national level the eligibility to the gene therapy treatment. Another solution could be the prenatal screening, which can identify the disorder before the birth, but this has to be discussed in all ethical frameworks. Second principle is the non-maleficence, so avoiding harm. It's difficult to talk about avoiding harm as a caregiver, but in gene therapy there is a double phase. It's a great treatment because we can give a solution for incurable diseases, but on the other hand, there are some difficult points to resolve, which are linked to the complexity of genetic disorders treated by gene therapies, but also to the technical problems linked to the treatment, which are the delivery challenges and the safety concerns, especially the risk of insertional mutagenesis, the immune reaction to gene therapy, and the potential long-term side effects. For the potential long-term side effects, I would like to talk to you about the severe combined immunodeficiencies, and the first trial conducted in a care hospital by Professor Cavazzano and Professor Fischer in 1999. Severe immunocombined deficiencies is a life-threatening genetic disease affecting the immune system, and with the lentivirus treatment, we were able to treat 10 patients, and the results of the treatment was quite successful because all these patients presented a T compartment reconstituted. But 6 out of these 10 patients presented a secondary leukemia linked to the gene therapy treatment because the lentivirus used at that time, the gene therapy product, was not so specific and was, there was an mutational insertional link close to the protoncogenes, which drive the malignant transformation. And it is important to note that the last leukemia has been developed 18 years later after the gene therapy treatment. For another kind of safety concern with the gene therapy is the presence of genetic variants of unknown significance in sickle cell disease. It is known that patients with sickle cell disease present a higher incidence of leukemia, of myeloid leukemia, because they could present at the bone marrow some clonal hematopoiesis. But in these cases, the gene therapy is not the only responsible of this transformation, but there are several steps and several hits, like the chemotherapy conditioning regimen and the gene therapy itself, which are able to stress this clonal hematopoiesis and induce a secondary leukemia. So how we can overcome also this problem? In our center, we perform now an NGS analysis, or a genome-wide analysis, in order to detect pre-gene therapy the presence of clonal hematopoiesis and to discuss later the eligibility of these patients to the gene therapy. Fourth ethical principle is the autonomy, respecting patients' decision. I know that we have already discussed about it. It will be discussed later. But let me say that it's very important to give the right information to patients and to their family before a gene therapy treatment, because this is true for all the drugs, but especially for gene therapy, because we have a complex scientific information that could be very difficult to be understood. And we have to take in mind that sometimes we have, in my experience, I have pediatric patients, so adolescent and young adults, and sometimes the opinion and the decision of the minor patients is not the same of the parents. So we have to take into account the importance of the information for patients and their family, and also to consider the ethical consideration in different sociocultural contexts. The last is not the less important, and this also will be discussed later, is the justice, the fair distribution of resources and treatments. As you know, and as my colleague underlined before, the development and the production of gene therapies is still very expensive, and this determines a high cost of gene therapies, and then an accessibility and affordability of gene therapy, which is not the same all over the world. This topic, this problem, has been already discussed several times from the scientific community, but at that time, the gene therapies are still very expensive, and the accessibility to other to all the countries in the world is not the same. For instance, one example is the treatment for beta hemoglobinopathies, beta thalassemia, and sickle cell disease. We performed the first clinical trial sponsored by Bluebird Bio, and thanks to the great results performed in the phase 1/2, these drugs has been the trial has been conducted also in the phase 3, and now is on the market. But the elevated cost proposed to Europe didn't allow the market of these drugs in Europe. So our patients are still waiting for this kind of treatment, especially for beta thalassemia, since for sickle cell disease we have the gene editing strategy on the market. At that time, we performed a cost-efficacy analysis in order to determine if the gene therapy treatment for beta thalassemia was more expensive than allogenic stem cell transplantation. And as you can see here, yes, the gene therapy treatment at that time was more expensive, especially for the production of the viral vector. But now, with all the new strategies that I showed before, we can imagine that the costs are less important, and moreover, we have to consider that the follow-up of patients treated by gene therapy is less expensive than the follow-up of patients treated for an hematopoietic stem cell transplantation with some different kind of toxicity. So in conclusion, while I have not all the answer to the critical questions of on ethics in gene therapy, we have some issues that we have to address at the governmental and private sector in ethical governance, while there are some issues that have been addressed and have been resolved by healthcare professionals, and these questions concern the safety assessment, the information given to the patients, and also the capacity to perform a collective decision-making process. So these are some suggestions. Thank you for your attention. I would like to thank you, my mentors, Professor Cavazzana, Professor Treluyer, and Professor Fischer, and I thank for sure all the patients and the family for their help in thinking about what we can do to improve our work. Thank you.
Thank you very much, Michaela. Thank you. Michaela, thank you very much.
Arnold, would you like me to address that question, or would you like to make other comments.
Thank you very much to Michaela for a superb talk. If you see hand raised, I'd love to have the first questions to Michaela first, maybe.
Let me just while we're waiting for a question, let me just address the question that was out there from before. So
Okay.
yes, we are the same CSL.
Okay.
And we have relentlessly and methodically diversified from just a plasma and flu company, and happy to talk to you about that story over a drink sometime, or my colleague Greg can give you some reference material, what we've published about that. Secondly, your question, yes, it was Jesse Helsinger was his name, in the University of Pennsylvania, and it's a sad story about, I think, not appreciating the law of unintended consequences. And Michaela talked about that in non-malfeasance, is the hyperreaction to a subunit of the AAV is probably what caused his demise, and it's a really sad story, and it probably pushed back gene therapy at least 15 years. And I know I have a colleague who went to the FDA about a week after and was told, you won't be coming back here for a decade. And she had a very promising therapy, so that's what it did. So it just reminds us that while we all believe we're doing good, we have to remind ourselves that there are other downside. And we, in fact, stopped the sickle cell program ourselves because my researchers saw something concerning, and we thought it was a possibility of the same kind of harm that Michaela was talking about, and we stopped that program. And I think we have to always remember that.
Thank you very much, William, for addressing this question and in a very gentle manner. Thank you for that. Do we have questions for Michaela?
Yes, it's out front.
Yes, sir. Yes, sir.
Yeah, go ahead. Thank you.
I'm a medical doctor and working with Cube Labs. So the questions related to pharmacoeconomy, I mean, the process of approval of cost of quality, which is always the problem with HTAs, in order to accept reimbursement for this kind of therapies. So I would like to know your opinion how you handle this, because this is a pretty solid barrier in some of the markets to get into the markets. And one comment on the side, I'm old enough to remember the phase of cystic fibrosis treatment. And when I was working with the company, we were saying that there was an era before the pancreatic enzymes, like a new technology, new Creon, and after. And we see dramatic improvement of quality of life of these patients. And then there's another one, when there's this CFTR gene modifications, and there is another longevity, and the quality of life of these kids are improving. So I'm happy to be witness of this kind of a change in medicine. However, still I know that a lot of therapies are actually being blocked after registration on the European Union soil, that they are facing problems with the reimbursement and HTAs, and the cost of quality is actually prohibitive. So what is your policy on that?
It's very difficult. Cystic fibrosis is a unique situation, because at the moment we have a fantastic drug. It's non-gene therapy. Now gene therapy is completely down now at the moment. I'm working very large time, so spent a very large time on cystic fibrosis. But what about the other diseases? For other metabolic disorders, we have it's not very, it's difficult, because we need to understand better the biology and the biochemistry. For this reason, I'm stressed in my lab, other lab, to spend many times to look at organoids from patients. So you take cells from patients affected by specific disorders to transform in iPS and differentiate in specific cells type or tissues in order to perform gene therapy and drug selection in this cell type. This is important. This can avoid the gene therapy approach, because you can also found some small molecules or others, for chemicals, for example, in order to test what about the production. It's different by the gene, because you have a defect in the mutation, you have a gain of function or
May I add something?
Yes, thank you.
Because You're right. Cystic fibrosis is a great example because there is a big gene involved with the several kind of mutations, and from an ethical and socioeconomic point of view, for sure the modificator of the CFTR, the production of the vertex drugs, modifying completely the life of the patients. But these are very expensive drugs, and you have to take these drugs, the patients have to, has to take these drugs each day. While this is the reason why the idea of a gene therapy for cystic fibrosis is still probably a possibility, because you can change the genetic disease itself and modifying the courses of the disease. But there are some trials on gene therapy on cystic fibrosis, but we will see the results.
Any other questions?
I think
Because we are running out of time, so is there any further question?
I don't think so.
Yes?
No.
So you can just stop with the time.
Oh.
Chair?
Yes.
Arnold, I think there's no more questions in the room.
Okay. So thank you very much, dear friends and colleagues. I think that we have had a superb session. And what I want to share with you in the next couple of seconds is that the advance in gene therapy cannot be the privilege of the few, as all of you have said. The advance, the actual progress is not the progress for the few, it's the progress for everyone, and how we organize the sharing of the progress. And thank you to the three of you to have addressed this question of how we shall move from a treatment for a few to a treatment for all, because it was a leitmotif of your three talks. And I want to express our deepest thanks for this major ethical issue, is to organize the sharing of our advances in gene therapy. So thank you very much for all of you. Thank you for your questions. Thank you to the audience. And I wish you an excellent meeting. Good afternoon.